Welcome back. This week's experiment explores the process of osmosis. This is how water molecules move from areas of high concentration to low concentration. The walls of plant cells have semi-permeable membranes, and through osmosis, this allows the cells to either expand or shrink depending upon the direction the water moves. This is how the roots of plants absorb water from the soil. But osmosis can be harmful to certain organisms in certain habitats. For example, if you took a saltwater fish, such as a clownfish, and put them in a freshwater environment, it would quickly die. And the same in reverse. If you took a freshwater goldfish and put it in the sea, it would also quickly die because of the osmosis effect on its cells. In this week's experiment, we're going to look at how osmosis impacts gummy bears. To run this experiment, you need 3 beakers, one with 2 teaspoons of salt, one with 1 teaspoon of salt, and one with no salt at all. You then need some fresh water, such as tap water, a set of scales, a ruler, 3 nearly identical gummy bears, and a results sheet, which you can download from the DARWIN200 website. Okay, let's get started. First, let's make our solutions. Pour in exactly the same quantity of water into each of the 3 beakers. I'm going to put in exactly 75 millilitres into each of these beakers. And the last one with no salt at all. Make sure you dissolve the salt. Mix the solutions very carefully. Make sure you also make a note on each beaker of the different solutions. So I've put a big S here for the solution with 2 teaspoons of salt, a small S here for the solution with only 1 teaspoon of salt, and no salt at all for the beaker containing the pure water. Then you're ready to weigh and measure your gummy bears before the experiment. So use a set of scales and very carefully put on a gummy bear. This one weighs 2.2 grams. I'll put that in front of the no salt solution. This one weighs 2.3 grams. I'll put that in front of the weak salt solution. And this one weighs 2.3 grams. I'll put that in front of the more concentrated salt solution. You then need to measure the length and the width of your gummy bears to see how they change as a result of your experiment. This one is 21 millimeters long and 12 millimeters wide. So make a note of that so you can compare your results afterwards. This one is 22 millimeters long and 11 millimeters wide. And the last one is 22 millimeters long and 12 millimeters wide. So we'll make a note of that too. So they're all very close in weight, length and width. Now put 1 gummy bear in each beaker. So put 1 in the fresh water, 1 in the weak salt solution, and 1 in the much more concentrated salt solution. You then have to leave your experiment for 24 hours. Come back after a day and see how each of the gummy bears has changed. Note down what changes you observe. Which gummy bears have grown bigger or smaller, and why? And how does this relate to the concentrations of salt in your beakers? Send in your answers to classroom@darwin200.com or upload your results via our website, www.darwin200.com. Please join us in 2 weeks and we'll find out the results and the reasons why these gummy bears change.